So today I'm going to talk about some simulations that we've done on blue waters and simulating whole cells. So the kind of simulations that I do are a little bit different than what we've heard about today. Generally, I think pretty much everyone in here is familiar with molecular dynamics simulations and probably to some extent Brownian dynamics. But when you want to start looking at whole cells, we have to coarse grain the representation even further than even Brownian dynamics. So what we are using for our simulations is a reaction diffusion master equation based approach. So what we do is we treat all of the chemical species in the system as point particles, and we're only interested in their diffusion and their reactivity between other particles. So in taking this sort of a coarse-grained approach, we're able to hit really, really long timescales, up to like 5 or 6 hours. Length scales, we can hit pretty large length scales right now as well, up to a couple tens of microns. So let me give you a quick little rundown of how you would set up one of these simulations. So the idea is you have some sort of geometry that you're interested in simulating. Here I have a cell with a membrane, a nucleoid region where the DNA is found, and cytoplasm. And then there's a bunch of species in there, like maybe metabolites that can come in, or you have DNA specific sites in here where translation or transcription occurs. You have all this set up, and to do the reaction diffusion master equation simulations, you discretize the whole thing down to a lattice. And each lattice site takes into account the number of species that are present. So this site here would say that there's 1 of whatever the black species is and 0 of each other species. So these species can then hop between nearest neighbor lattice sites, or if there's more than 1 species there and they're able to react, they can react with each other and create or destroy species. So the dynamics, like I said, is governed by the reaction diffusion master equation. And what this equation does is it specifies the probability to find a particular state of the cell. So the first term here governs the reactions. So this is basically just telling us the probability flux due to reactions out of the state plus reactions into the state. And the second term here is probability flux due to diffusion. So just what exactly do I mean by the probability of the state of the cell, this X vector here? Well, in terms of these simulations, what it corresponds to is terms that are given for each lattice site. And each one of these lattice site terms here tells you the number of particles for each species type that we're simulating. So the software that we use is called Lattice Microbes, and it's developed by the Luthi Scholten lab. We designed it from the ground up to use CUDA. So each particular lattice site in the simulation is controlled by a single thread on the GPU. And we went beyond just a simple general purpose GPU parallelism, and we've been able to split the simulation volume across multiple GPUs. And currently we're working on an MPI implementation that'll split it across the entire supercomputer. So one thing that's nice about the way our software is designed is it's completely extensible through Python. So if you're interested in setting up some sort of interesting, weird simulation setup that we didn't intend initially, you can just use the Python interface to design those simulations on your own. So the problem I'm interested in is the biogenesis of the ribosome in E. coli. So clearly the ribosome makes protein in the cell. It's really important. It's composed of the large subunit, which is made up of about 50 some odd proteins and 2 strands of RNA, and the small subunit, which is made of about 20 proteins and 1 strand of RNA. So altogether, this takes up about 1/4 of the cell's dry mass. If you're interested in coming up with a whole cell model of E. coli that includes ribosome biogenesis, for ribosome biogenesis alone, you need to understand gene regulation. So how the ribosomal RNA is translated or transcribed, and how the protein mRNA is transcribed. You need to know how, or regulated rather, you need to know how transcription works, and you need to know how translation works. So by the time you figured all of this out, as well as the ribosome assembly, you're at a really, really good point because you have the majority of the whole cell model figured out. So from here, you can add more complex models to the whole cell system, such as metabolism or other cell signaling type networks. So the assembly of the small subunit is kind of complicated. So like I said, there's one strand of RNA and 20 proteins. Each of these proteins can bind to this RNA here, and then there's a handful of cofactors that enable the assembly in vivo. So I'm using lattice microbes, so I'm not super interested in the molecular details. So that seems to simplify the problem somewhat, but not really, because if I have 20 proteins binding to a single substrate, that's 2 to the 20 intermediates and 20 factorial reactions that I have to deal with. So we have to simplify this quite a bit before we can continue. The way that we chose to simplify it is to use an experimental observation from Nomura back in 1970, I think. And what he found is there are a couple of classes of small subunit proteins. The primary proteins can bind directly to the RNA, whereas the secondary proteins can only bind if there's a primary present. So for S16 to bind, we have to have S4 and S20 already, but S17 can bind on its own. So what we can do is we can then enumerate all of the possible intermediates and all of the possible reactions. And if we do that, we end up with 2,600 reactions and 2,600 species and about 6,000 reactions. Okay, so that gives us the network topology, but we're interested in dynamics here. So for the dynamics, we get this from a pulse chase quantitative mass spectrometry study. And what they have done is they have basically assembled the small subunit of the ribosome in vitro. They take the RNA and they add a bunch of radiolabeled proteins, and then they let it react. At some point in time, they add an excess of non-labeled proteins, let it finish assembly, and then compute the fraction of radiolabeled proteins to unlabeled proteins. And that gives us these protein binding curves. We can then come up with a kinetic model using our assembly network and then extract rate parameters. So now that we know the rate parameters, we can even simplify it further, because if we run the simulation out or run the chemical equations, the chemical rate equations out, we can see which of these assembly intermediates are actually being used by the network. So what we did is we actually removed the majority of them simply because there was no flux through those intermediates. And what we ended up with is about 147 intermediates and about 400 reactions. So the nice thing about this model is it actually predicts intermediates, like exact intermediates to see in experiments. And it turns out the ones that are in boxes here have been observed using cryo-electron microscopy. And it also predicts the right assembly order, or at least the assumed assembly order that people have seen so far, which is a kind of a 5' to central domain to 3' directionality. Cool. So that is the assembly model, but ribosome biogenesis isn't just assembly. It has to take into account the transcription of the ribosomal RNA, the transcription of the messengers for the ribosomal protein, as well as degradation of the messenger and translation of those proteins. So this data has to come from somewhere. So some of this data we can kind of just assume. We know based on certain experiments on E. coli that there should be about 6,000 prior to cell division in slow-growing E. coli. So we choose transcription rates that achieve this quantity of ribosomes at the end. We choose kind of we don't know exactly what to choose for the messenger, so we just ensure that there's an even abundance of all the ribosomal proteins and about a 50% excess. Degradation comes from microarray data, and the translation rates just comes from a bio numbers measurement of 10 amino acids per second and the transcript length of the messenger that it's translating. Finally, diffusion constants are either estimated or based on the radius of gyration, or they come from single particle diffusion experiments of either the ribosome or the ribosomal subunits. So I'm going to walk you through a simple little movie here, which shows the basic layout of one of these simulations. So it's a lattice, and you can see that it's the simulation volume is a box essentially. Inside the box, there are a number of different reaction compartments There's the exterior, there's the membrane, there's the cytoplasm, and then there's the nucleoid region where the DNA is found, and typically ribosomes are excluded from that region. So in the nucleoid, we place sites where transcription can occur. So we have R protein operons, which produce the messenger, and ribosomal RNA operons, which produce the ribosomal RNA. And these are placed along the z-axis of the cell such that their position reflects their position in the genome. So at these sites, or at the sites of the ribosomal protein messengers, mRNA are produced. And they're produced at a constant rate since we don't have gene regulation currently in the model. And these can then diffuse around the lattice by hopping to nearest neighbor lattice sites. And then translation can begin through the binding of the mRNA with the small subunit. This new complex can then bind with the large subunit, forming a translating ribosome. And just to rub it in, we've completely coarse-grained that to a sphere, because, you know, I'm a physicist. We have, so the translation process is then goes according to the transcript structure. So we start at the beginning of the transcript, and then the time that it takes to produce the first protein is given by the transcript length of that protein. Okay. The next protein is produced by considering the position of the ribosome currently and the end of the next gene to produce. And finally, to degrade the ribosome, it translates to the end and then falls apart into its three constituents, which can then also participate in a translation again. So assembly begins by the production of ribosomal RNA from one of these operons, and then this ribosomal RNA can now diffuse around the system, picking up proteins in vaguely the order of the Nomura map. And once it has assembled completely, it forms a 30S small subunit, which can then participate in translation like everything else. So one new thing that we've added to this model recently is cell division and DNA replication. So in E. coli, or in most bacteria, I guess, there's typically three portions of the cell cycle. There's a B period where there's no gene replication. There's a C period where DNA replication is occurring. And then there's a D period, which is a period that it waits before it divides. So I have made I've made an assumption here. And that assumption is only one round of DNA replication occurs at once. Now that's true for slow-growing E. coli, but for fast-growing E. coli, you can have multiple rounds of replication, and that will make the simulation quite a bit more complicated. So we're going to stick with slow-growing for now. So Tom Kuhlman in the physics department of University of Illinois did some really cool experiments with E. coli. So he used slow-growing E. coli, and then he marked 14 different places in the genome, 14 different strains, each with its own portion in the genome, these yellow marks here. And he made it so that you can see these fluorescently under the microscope. So from this data, from these cells, we can simultaneously measure their length and the number of copies of that particular gene in each cell. So using that data, we can fit it to a very simple probabilistic model and extract the cell cycle parameters and the mean cell size at division. And what we end up finding is that the mean cell length at division is 4.7 microns, and the initiation time, the replication time are both about 40 minutes. So we can then take these parameters and use them to build our whole cell simulation. So for the geometry, we just use that time that we measured. In this case, it's going to be 2.4 initially, and then it's going to go to 4.8. And the width of the cell we can measure from our experiments as well, and that's about 0.7 microns. So the position of the nucleoid in the cells is also pretty important to know, because the ribosomes are excluded from the nucleoid region. So these little black dots here are ribosomes, and this is a cryo-EM tomogram of slow-growing E. coli. So we can measure these distances here and use them to assemble our whole cell model. So This is basically how the cell grows as a function of time over its 120-minute cell cycle. So these areas here in the different colors are the different compartments, and these lines show where the ribosomal operons are added. So what you can see here is the cell begins to split off the nucleoid region at about 90 minutes, and then it's fully divided at 120 minutes. So the volume of the cell and as well the length of the cell increases exponentially over its growth cycle. And what that change in volume does is it affects the concentrations. So the concentration of ribosomes in the cells, the explicit number starts at about 3,000 and ends up at 6,000. But thanks to the changing cell volume, it stays relatively constant. And you really only see an inflection point where the operons are duplicated. Okay, for protein, it's the same sort of deal. You only really see an inflection point where the ribosomal operons are duplicated, and it stays relatively constant over the whole cell cycle. So the proteins themselves can be found pretty much anywhere in the cell. So what I'm showing here is a projection of the particle density onto the y-axis, or onto the xz plane. So if you were to imagine that these proteins were fluorescently labeled, you would be seeing essentially what you would see in a fluorescence microscope. So the proteins can be found everywhere, but the ribosomes cannot. They are partially excluded from the nucleoid region. So you can see that they're found typically at the end caps and around the cell membrane. Finally, the ribosomal intermediates, these are relatively short-lived, so they tend to kind of accumulate near their operons in which they were born from. So they kind of just follow the trajectory of the operons. Okay, so here's a typical simulation. I have labeled, in addition to the other operons which I labeled prior, I have labeled the translation initiation events and the positions of all the intermediates. So you can see translation initiation can occur in a couple of locations. It can occur near these operons, or it can occur outside the nucleoid region. And basically what's happening here is the small subunits can enter the nucleoid region, but the ribosomes tend to diffuse out and stay out. So you see translation initiation occurring only near the operon sites and outside the nucleoid region. However, for the assembly, that tends to happen pretty much everywhere in the cell. Okay, so I'd like to talk a little bit about how fast this stuff is. Prior to some work that one of my colleagues, Mike Halleck, did, these sorts of simulations were really difficult, simply because we have a large number of reactions. We have about 900 reactions when you consider all the different compartments involved. And what that leads to is a wall time per time step of about 22 milliseconds. So the time step in our simulations is 25 microseconds. And we want to do 2 hours, so that's a lot of time to simulate an entire cell cycle. So he came up with a different reaction computation technique where it only loops over the particles that are available in each lattice site. So doing that, we cut it down to about 2 milliseconds on Blue Waters. Finally, I'd like to go over a little bit of benchmarks we've done on another GPU, which is a Titan X. We're actually, it's really impressive how fast this thing is. We can get it down to about, I think it's 800 microseconds per time step, which is pretty impressive, which then leads to about a day to simulate an entire cell cycle. Whereas on Blue Waters, we're looking at about a week. But that doesn't mean that I don't use Blue Waters. I use Blue Waters a lot. If we want to do any real sampling, we need to use Blue Waters simply because we don't have a bunch of these gamer boards hanging out in our lab. Secondly, though, one of the other things that we plan to do with lattice microbes is we intend to look at really large systems. So once we have the MPI implementation completely figured out, we're going to want to look at stuff like eukaryotic cells, bacterial colonies, and tissues. So in order to do that, we will definitely need blue waters or a system similar to it. So the remaining things to do in my E. coli model is basically I want to bring in regulation, so I don't have to kind of pull those rates out of thin air. An explicit treatment of the DNA, which I hinted at on my title slide with the tangled up DNA. And we want to actually bring in metabolism to have a full cell model. So in lattice microbes, I mentioned we wanted to do MPI. Well, the other thing that we want to do is a multi-scale modeling approach. So when you have something like ATP, that can be found in concentrations of like a couple of millimolar in the cell. And it's a small molecule, so it diffuses really quickly. So treating it with a fully spatially resolved stochastic approach is kind of a waste of resources. So we'd like to be able to use an ordinary differential equation, chemical rate equation approach when necessary, or a non-spatially resolved stochastic simulation when necessary, or just a PDE type approach when necessary, just solve the reaction diffusion equations. So I'd like to thank my advisors, Ann Schulten. Tom Kuhlman in the physics department did all of the experiments. Mike Hellick is an amazing research programmer who works with us. And Joe Peterson and John Cole also worked on this project with me. If you're interested in lattice microbes, you can Google it, or you can go to this tiny URL link I prepared for everyone. Thanks.